replication occurs in a variety of steps and require various enzymes to complete this process. As you can see here, the two strands of DNA, along the backbone you have a phosphodiester bond, and in between the two strands that are running from 5' to 3' and 3' to 5', which are anti-parallel, you have hydrogen bonds between A and T, which are adenine and thymine, and G and C, which are guanine and cytosine. There are 2 bonds between adenine and thymine, and 3 bonds between guanine and cytosine. The first major step for the DNA replication to take place is the breaking of the hydrogen bonds between bases of the 2 anti-parallel strands, running from 5' to 3' and 3' to 5'. The unwinding of the 2 strands is the starting point, and the splitting happens in places where in the chains where they are rich in AT, and that is because there are only 2 bonds between adenine and thymine, whereas there are 3 bonds and it is harder to break between cytosine and guanine. Helicase is the enzyme that is responsible for splitting the 2 strands, and the initiation where the splitting starts is called the origin of replication. So the helicase comes in and splits the strand, and this area right here is called the replication fork. The helicase is responsible for unwinding this strand. The topoisomerase is responsible for relieving the tension between these 2 parental strands. And then you have a protein called the single-stranded binding protein, which is responsible for stabilizing this unwound parental strand. Again, hydrogen bonds were broken between the adenine and thymine because there were only 2 bonds between those 2. And the helicase came in. It allowed for there to be a splitting between the parental strands. The topoisomerase relieves the tension between this strand to prevent supercoiling. And then you have single-stranded binding proteins, which stabilizes the unwound parental strand. One of the most important steps of DNA replication is the binding of the RNA primase in the initiation point of the 3' to 5' parent chain. RNA primase can attract RNA nucleotides which bind to the DNA nucleotides of the 3' to 5' strand due to the hydrogen bonds between the bases. And RNA nucleotides are the primers or starters for the binding of the DNA nucleotides. And the RNA primers are what are the starters for the binding of the DNA nucleotides. This complex right here is called the RNA primase and is attached at the 5' end of this DNA strand. The elongation process is different for both the 5' and 3' and 3' to 5' template. In the 5' to 3' template, the daughter strand that uses the 5' to 3' template is called the leading strand. And this is because DNA polymerase can read the template and continuously add nucleotides that are complementary to the nucleotides of this particular template. So because DNA polymerase comes in, it is able to continuously add the nucleotides. So DNA polymerase 1 is responsible for adding the nucleotides that are complementary to the nucleotides of the template. In the 3' to 5' template, it is harder for the DNA to be read by DNA polymerase, and that makes the replication of this particular template more complicated, and the new strand being added is called the lagging strand, as seen here due to the discontinuity of this strand. This is the lagging strand. In the lagging strand, the RNA primase adds more RNA primers, as seen here, and DNA polymerase 1 reads the template and lengthens the burst. And the gap between the RNA primers is called the Okazaki fragments of the lagging strand due to the discontinuity. The RNA primers are necessary for DNA polymerase 1 to bind to the nucleotides to the 3' end of the chain. DNA polymerase 3 is also responsible for coming in to close this gap of the lagging strand so that it can be continuous. and nucleotides are added. In the lagging strand, the DNA exonuclease reads the fragments and removes the RNA primers. So this is the DNA exonuclease, and it comes in to remove the RNA primers. The gaps are closed with the action of DNA polymerase, which when the DNA polymerase comes, it will add complementary nucleotides in these gaps. And these nucleotides that I'm adding are due to the DNA polymerase. The DNA ligase, which is another enzyme, comes in and adds phosphate in the remaining gaps of the phosphate sugar backbone. And this represents the DNA ligase, which will come in and joins the sugar phosphate backbone of the Okazaki fragments to form a continuous strand. Each new double helix is consisted of one old and one new chain. And this right here is called the semi-conservative replication because it has one old and one new chain. And then these represent the removed primers. The last step of DNA replication is the termination step. And this process happens when the DNA polymerase reaches to an end of the strands. So this is the DNA polymerase. It reaches the end of the strands. And we can see this and understand this as explained in the last section when I described the lagging strand when the RNA primer is removed. It is not possible for the DNA polymerase to seal the gap because there is no primer. So the end of the parental strand where the last primer binds isn't replicated. These strands of linear chromosomal DNA consist of non-coding DNA that contains repeated sequence that are called telomeres. As a result, a part of the telomere is removed in every cycle of the replication. The DNA replication is not completed before a mechanism of repair fixes the possible errors caused during the replication enzyme. These enzymes are called nucleases, and they remove the wrong nucleotides, and the DNA polymerase fills the gap. So because right now the polymerase cannot fill the gap because the primers aren't present, when the nuclease comes in, it removes the wrong nucleotides. A and G do not go together. They are removed. Exonuclease also removes C and T. They do not go together, so it is removed. The polymerase allows for the correct nucleotides to be placed. And then the ligase will seal this gap. You now have a continuous strand, two continuous strands of DNA double helix strands running from 3' to 5', 5' to 3', and again 5' to 3', and 3' to 5'. The processes happen all during the steps of DNA replication.